Hello, today we're going to talk about the synapsis of these two chromosomes. So the first one is MIJ centromere KLHG, and the second one is MLK centromere JIHG. So we have a pericentric inversion causing a loop over here. So I made this diagram using cables and my organic chemistry molecular kit. So the first strand that we have is MIJ centromere KLHG, which is a viable one. The second one is MIJ crossover
Centromere
KLM. So we have two copies of the M gene, so it will not be viable. Our third one is MLK centromere JIHG. So this one is viable because we have a single copy of every single gene. And then the fourth one, we're going to start from here, is GHLK crossover
centromere
JIHG. So this one will not be viable either because we will have two copies of H and G. Our second set of chromosomes will be MIJ centromere KLHG with GHLK centromere JIM, which when we inverse the other one, we realize that they're equal to each other. Therefore, we have no loop and we just have this. So the first strand will be MIJ KLHG, which is viable. Second one is MIJ crossover centromere KLHG. Third one is MIJ crossover centromere KLHG. And fourth one is MIJ centromere KLHG. So all four of them are viable. Individual 2 has 4 viable gametes, while individual 1 only has 2 viable gametes. Therefore, individual 2 will have better fitness.